talking about epithelial cells, basement membrane. What's the difference between a basement membrane and a basal lamina? When you get something like a blister, this is where some irritation, maybe brand new shoes, maybe some heat that you came in contact with that caused fluid to leak out of your vascular system and into the tissue and separate the epithelial cells from the underlying tissue from the basal lamina. So that fluid buildup is what you have as a blister. Do not pop blisters. Your epidermis is there to protect your underlying tissue from bacterial invasion. If the blister pops, then bacteria gets in there and it could just cause a local infection, or it could streak all the way up your arm, or it could go in your bone and create a real nasty mess. So as long as possible, do not pop the blister. When the epidermis heals from underneath, the blister will pop on its own and the fluid will come out and it's fine. If you accidentally pop the blister, make sure you put antibiotic on it, like bacitracin or neosporin or something. Keep it clean. Burns, they're different. These are the, I guess, kind of the old terms. There's actually a 4th degree burn, and they have different classifications now, but in general, 1st degree burn is where it's red like a sunburn, but if fluids collect under there, you're going to get a blister. A 1st degree burn doesn't always have a blister, but it could have a blister. You start to exercise and your sweat glands can't empty onto the surface anymore because the epidermis is separated from where the sweat gland is secreting. You're going to get a blister because of the sweat that's coming out of your gland. Okay? But then you could have a 2nd degree burn where you really have a blister because of that degree of damage. So you have death to some of the dermis, but you haven't gone all the way through the dermis. Thank goodness, because you have sweat glands that invaginated from your epidermis, and they have the same stem cells that will help to replace the epidermal cells. Also, intact epidermis cells will migrate in and help to heal. And I should bring in the model that one of my students made of damage to the skin. I'll bring it in when we get to the skin chapter. But she took it stage by stage, her right brain project. And she had the damage, and then she had the healing process, and she had all her little cells and different little sparkly thingies. And it was fun to look at, and it was also very educational. So 2nd degree burn, you damage the dermis. Now you not only have to replace the epidermis, but parts of the dermis. But since surrounding areas, hopefully, are still intact, you get the stem cells undergoing mitosis, and they'll migrate into the area and make the repair. And again, your hair shafts, your sweat glands, they're all from the epidermis anyway, so more stem cells to help you repair. But if you have what's called a full thickness burn, that means you've gone through the dermis entirely down to the subcutaneous or even to the muscle layers, then you don't have the hair shaft anymore, you don't have the sweat gland anymore, you don't have the epidermis obviously anymore. If you destroyed the dermis, you don't have epidermis. So it's going to take longer for those types of burns to heal. You might actually, there's a lot of new techniques for helping burns heal. It used to be you'd have to graft it or get cadaver skin or pig skin, or if you had enough of your own skin, they could take the skin and make little slits in it and stretch it, and then the cells would granulate and come in and fill in. So you had, those were the old techniques, and now they can take your stem cells and grow it in the lab and bring it back to you. They have a lot of marvelous things. But 3rd degree or full thickness, sorry, press the wrong button again, burns, they're through, usually into the hypodermis, which is your fat layer with blood vessels and other things there, but primarily fat, and some connective tissue into the muscle, or could be all the way down into the bone. So generally when it's invaded invaded muscle and maybe down into the bone, they sometimes now call it 4th degree burn. And that's the worst, because you not only have to repair the skin, but you got to repair other tissues as well. So cuts, they're usually minor. The neater your slices, actually more it bleeds. So when the surgeon goes in there with a nice scalpel and makes a nice clean cut, you have to follow that with cauterization. And if you have a ragged cut, a laceration, depending on how lacerated the tissue is, but a more jagged cut, it may scar, but it actually heals better and bleeds less than a nice neat cut. But depending on how deep you cut, we're going to talk more about the response of your body to cuts later on. But if you were running your hand along the paper and got a paper cut, you go, ow! And you look, nothing to worry about, no bleeding. And then all of a sudden you look down and there's blood all over the paper and you're just oozing all over the place. That ow! Vasospasm prevents the bleeding. But as soon as that is relieved, then you start to bleed. So you get and then you get a second spasm later on due to the chemicals of the blood clotting. So we'll talk more about that probably next semester. But these are all damage to epidermis, which is your epithelial cells. You've gone through the basement membrane, or you've separated the epithelium from the basement membrane. And now you've got to heal those things. So here we start to look at other types of epithelia. Epidermis is stratified squamous, plus it's keratinized for increased reinforcement. So things like blisters, you get blisters in your mouth when you put hot food in there, and those blisters pop very easily. Why? Because the tissue is not keratinized. So any little pressure on those cells, they just come apart and the blister pops. Blister on your skin is a little bit more resilient. Back to columnar. We saw those columnar cells on the first epithelial slide. It's taller than it is wide, so by definition it's columnar. Epithelial cells, depending on where their location is, they're going to be having their apical plasma membrane modified based on what function they need to provide. So we've got all these columnar cells, and some have stereocilia, some have cilia, and some have microvilli. So actually stereocilia, means non-motile. So cilia, they are whipping, they have a whipping action. Stereocilia are the same thing as a microvillus. But they're categorized separately because these stereocilia are very long. If you look at the illustration, stereocilia are even longer than cilia. When you look at the core of a microvillus and you look at the core of a true cilium, one has microtubules, that's the cilia, and that allows for the whipping action and with other motor protein. The core of a microvillus is going to be actin. So microvillus will have actin, which is your thin filament or microfilament. Your true cilium, singular, or cilia, which is plural. This is going to have a basal body, which is made from centrioles. Well, acentriole, so take the S away. This is going to be microtubule. As opposed to actin. So the core is going to be made of something different. It's going to appear different. The basal body is going to have 9 triplets. So 1, 2, 3, 4, 5, 6, 7, 8, 9. 1, 2, 3, 4, 5, 6, 7, 8, 9. So 9 triplets. I'm just not going to draw them all. 9 triplets in a circle. Microtubule structures above the basal body. They're going to have doublets for most of the 9 doublets. And then 2 more in the center. So they look, the upper part of the cilium will look different than the basal body. But this structure will form that structure and then push the plasma membrane up. What is the function? Stereocilia, if they're microvilli, then these are going to be involved in absorption. They're going to absorb or secrete. So absorption and secretion. Increase the surface area for absorption and secretion. The actin is there because it will interact with other filaments down in the cell and kind of milk what's being absorbed down into the cell. It also will interact and kind of stir up the fluid that's over the microvillus, so you don't deplete the fluid of nutrients that you're wanting to absorb. So actin is not there to actually move anything over the surface, but just facilitate absorption. Stereocilia, you find those in the male reproductive tract, and specifically in the epididymis. Cilia, you're going to find those in your respiratory system, so in your sinuses, in your large conducting airways. You'll find a cilia as the tail of a sperm. It's actually a flagella in that case, but it has similar structures to a cilium. It's a little bit different in its function. Instead of whipping, it kind of acts as a rotor. Microvilli, come back. Cilia, you also find that in the female reproductive tract because you want to get the ova from the ovary, which is just ovulated. You want to get it into the fallopian tube and down through the fallopian tube, oviduct, or uterine tube. There are all these names we'll talk about next semester. You want to get it towards the uterus in a place where fertilization can occur safely. Don't want fertilization and implantation occurring out in the pelvic cavity because mommy will rupture, bleed all over the place, and perhaps die. The uterus is really the only place that's designed for implantation of a fertilized oocyte. So cilia and microvilli together, you find that in the oviduct. The cilia come and go throughout the cycle of the female. Towards ovulation, the cilia appear. When you've already ovulated, you're in the process of recruiting more, the cilia will disappear. You don't need them yet again, so the cells don't maintain them. The microvilli in those areas, they're doing more of a secretory function. They secrete a very sticky mucus that helps to trap the ova. not so sticky that the cilia can't move the ova, but sticky enough so that the ova will get trapped there. When you have columnar, so those are surface modifications that you find on a columnar cell. If you're doing secretion, you could be doing a secretion of some lipophilic substance, so it doesn't require a vesicle. But if it's something that has a protein component to it or a carbohydrate component to it, it's soluble in the cytoplasm, but it's not soluble in the plasma membrane. So to get it out of the cell as a secretory product, you've got to package it in a membrane that can fuse with the plasma membrane and then open up so the product inside can come out. So when you see the Golgi apparatus, always capital G because this is for an anatomist named Camillo Golgi. The Golgi apparatus is responsible for packaging things for secretion. Again, columnar cells will have their nucleus towards the basal side, towards the base, the basal lamina side of the cell. Mitochondria will sometimes be highly concentrated there. They might also be distributed up more apically. But if a cell is doing a lot of secretion, then the secretory products are going to be up here. They're taking up space, so everything else gets shoved to the bottom. It's just for convenience and probably working organization. It just works out better that way. To have the secretory products closest to the side of the cell where they're going to leave, and everything else on the other end. So mitochondria, what are mitochondria for? ATP production. Believe it or not, some people are going to leave this course and not know that mitochondria, at least on an exam, they're not going to know that it's the powerhouse of the cell. Totally amazed that didn't happen. So again, to emphasize the polarity of the cell, apical is going to be different than basal. Basolateral, sometimes things are very similar on the sides of the cell and the base of the cell. Anything that's absorbed apically usually can be secreted laterally and basally. So they talk about these two surfaces as basolateral surfaces, meaning functionally they're pretty much the same. Laterally, you'll have certain types of junctions that hold the cells together, and other junctions to hold the cells to the basement membrane or basal lamina. So yes, you have to know the locations of the epithelia. So study this PowerPoint well, be able to say on an exam, probably looking at a reproduction of the artist's illustration, because when I start to Xerox or duplicate that, it gets kind of mushy. But still, if you're looking at it and you know your epithelia, you should, even if this is blurry, you should be able to tell what you're looking at. So simple squamous, you look at it from above, it looks like a fried egg, has a centrally located nucleus, kind of irregularly shaped, round. Sometimes this is referred to as pavement epithelium. If you've ever seen a cobblestone in old English towns or other places, a lot of the cobblestones have this kind of irregular appearance, kind of dome shape at the top, but slanting down to where they attach to another cobblestone. The thinness of the cell when you do a section from the side shows you that there's very little distance from the apical surface to the basal surface. So this is good for increasing permeability. So you want gas exchange to occur, you want nutrient exchange to occur, you have a simple epithelium to decrease the distance that something has to diffuse. You get a thicker cell, you're going to have to do some more creative types of transport. But for diffusion purposes, simple diffusion or simple transport mechanisms, usually you put what's called a serous membrane on the surface of either, for example, the wall of the thoracic cavity, the inside of your peritoneal sac for your heart, your pericardial sac for your heart, on the surface of your intestinal organs. When you have two of these surfaces that are next to each other, the fluids that are coming from the blood on either side get secreted into the space between the two, primarily water. So the two surfaces tend to stick together, but they also can slide over each other. So when you breathe in, or when your heart changes shape, or when your intestines move, you get the organs slipping over each other. But in the case of the lungs, when your rib cage expands or your diaphragm goes down, then the two membranes move together, and that is functionally good for filling the lungs, because the volume goes up, pressure inside goes down, and as long as your glottis is open, air comes in. So when there are two layers together, they're there to reduce frictional forces. They can do absorption and secretion. So we'll talk about a process called transcytosis, which you absorb on one side and you secrete on the other side. So peritoneal lining, lining of the lungs, lining of the pericardial sac, these will all have simple squamous cell layer on the organ, and then also on the cavity wall. You get to stratified squamous, which we've already talked about. If you have nuclei all the way to the top, then you have non-keratinized stratified squamous. The cells are alive all the way to the top. So you find that in your oral cavity, nasal passage, your anus, your rectum, your urethra, and vagina. So if it's skin, you're going to keratinize it because it's exposed to the surface and frictional forces, your clothing, things you run into every day, your book pack, your books, and other things. So you don't want live cells at the surface. So the whole process of building the epidermis kills the cells, and then they're dead up to the surface. So you can afford to lose the top layers of your skin. You can lose some of these non-keratinized cells in your mouth. You scrape it on with your inside of your mouth with a swab for DNA. That's why you watch the TV and they say, "Oh, epithelial cells from the skin. Let's do DNA." You should just go. Those cells are dead. They don't have any DNA anymore. The nucleus is gone. But when they take a swab from the inside of the mouth, yeah, those cells are alive. They can do DNA on that. Okay, pharynx, back of your throat. There's a nasal, there's an oral, and a laryngeal pharynx, which then turns into the esophagus, still stratified, squamous, non-keratinized. Vagina, rectum, anus, stratified, squamous, non-keratinized. Urethra is not there, but that would have, the lower part of the urethra would have this type of epithelium closer to the surface of the tract. The external environment, you'll have the stratified squamous. These are designed for a lot of protection. This provides some protection, but again, just a piece of tissue and you touch it and move the tissue, the cells are then stuck to the tissue and removed from the surface. So they protect against abrasion, chemicals. You put the same shampoo or cream or deodorant that you put on your skin in your mouth, it burns. You don't want it there. You put it here in your, get it into your eyes, it burns because that's non-keratinized. So it's less resistive to chemical. It's not going to be as bad as if that same thing is touching a cell that, or touching an epithelium that has fewer layers, but it's still non-keratinized. You're going to feel it more than if it's keratinized. Cuboidal, as the name implies, it's a cube or relatively square shaped. It's as tall as it is wide. The nucleus, you come back and look here, the nucleus in squamous cell, simple squamous, is kind of equally located between the apical and the basal surface. Remember I told you last time, at least I hope I did, that when you name stratified epithelia, you always name it for what's on top. So you look down below, this is kind of cuboidal looking, and then it kind of gets weirder shape, starts to flatten out, and the top layers are all relatively flat. So this is stratified squamous. Even though you might have more cuboidal down below, you don't name it for what's down close to the basal lamina, you name it for what's on the surface. So here we've got basal lamina, different shapes of cells attached to the basal lamina. Got a straight duct here and probably a circular cross section through a duct up above. All the cells are about as tall as they are wide, and nucleus is in the center again. When you have a cuboidal cell, although it's not shown here, you have space for more organelles. Simple squamous cells don't have a lot of space for organelles. They're not going to have a lot of mitochondria, they're not going to have a lot of Golgi, they're not going to have a lot of other things to do a lot of function. So when you have a bigger cell, you can do more things. You can run more functions in those cells. Where do you find cuboidal cells? You find them in glands. Secretory glands like your sweat glands. Secretory glands that are secreting enzymes for digestion. These will have cuboidal cells that sometimes they kind of look more pie-shaped than cuboidal, but you'll find that type of shape in a gland. When you get out to the ducts, there will be transition, but a lot of parts of the ducts will have cuboidal epithelium. Kidney tubules, which are shown here. We'll talk more about it next semester, but how do I know it's a kidney tubule? Because of all the glycocalyx here. You're not going to have that in a sweat gland. You're not going to have it in a thyroid gland. Thyroid gland will look very different than that. It's going to be full of colloid. So eventually you're going to be able to recognize that as well. But for now, just memorize this. Kidney tubules and thyroid gland, and most ducts will have a portion that will have a long portion that will have cuboidal epithelial cells, and glands will have cuboidal epithelial cells. They provide more protection than a simple squamous cell, less protection than stratified squamous, less protection than a columnar cell. So protection is as the size implies or the stratification implies. But these guys with their glycocalyx, especially these that are really darkly staining as compared to these which are a little bit lighter staining, that dark staining means they have a lot more organelles and they're doing a lot more, they're functioning at a higher rate than these guys are. So these will be doing a lot more absorption and secretion. These are probably doing absorption and secretion, but only when they're told to by some hormone that comes by. Stratified cuboidal, as it says up here, very rare. You don't see stratified cuboidal in humans. It lines some ducts, but primarily it has disappeared from the fully developed human. So some ducts will have 2 or more layers. The top layer is cuboidal. Here, the bottom layer is relatively cuboidal as well. So you're still naming it for what's on the apical surface, which is cuboidal. The stratification provides more protection. You have limited ability to absorb and secrete. Why? You've got an extra layer. So whatever is secreted has to, the products or the building blocks for the secretion have to get through this first layer to the second layer, which then can secrete. Anything that's absorbing has to go through 2 layers to get to the bloodstream for distribution. So there's limited absorption and secretion there. It's found in a sweat gland duct, and this is primarily to send the sweat out to the surface. Some absorption and secretion occurs depending on the flow rate, so you can modify the sweat and not lose all the electrolytes. But a high flow rate, you're not going to get much altering of the secretion. Transitional epithelium is also called urinary epithelium. It's only found in the urinary system. So you're not going to look for it in the lungs, you're not going to look for it in the GI tract. It's urinary epithelium. It's called transitional because as you see here and here, the cells look different as they go up towards the surface. Very characteristic of this transitional epithelium, 2 big characteristics. The top cells have a dome shape to them. When you see them in slides in the lab, they will be very distinctly balloon shaped at the top. This allows the urine to collect in the lumen, stretch the epithelium, and the top cell layers don't rip apart. So you can sit there and collect a liter, a liter and a half, some people 2 liters of urine before they head to the bathroom. You're looking at me like, what? But yeah, sometimes you go into the bathroom and you hear somebody start to urinate, and it goes on and on and on and on and on, and you're thinking, my God, it's got to stop soon, and it goes on and on and on. It's not healthy to do that. You get a lot of urinary tract infections when you do that. But you have the capacity to override those stretch reflexes of a filling bladder, and some people choose to do that. But you're not going to lose the integrity of the epithelium because the top layers just keep stretching to accommodate that volume. Another thing that helps is that the basal lamina is incomplete. So in other areas, the fibers all interconnect, but there are gaps in the transitional epithelial basal lamina, so it stretches better and doesn't rip. So you've got epithelium that stretches, and you've got an incomplete basal lamina that also stretches. There is probably still controversy. Is this stratified or is this pseudo-stratified? Doesn't matter for this class. You can argue that in a graduate class. You're taking histology. For our purposes, this is either urinary epithelium or transitional epithelium. Or it will say urinary / transitional. Have you identify the transitional epithelium? You want to look for the dome-shaped cells on the top, and that will tell you you're in the urinary system. So where do you find it? You don't know probably, unless you remember from your early classes, what the major and minor calyces are. But that's where urine that has been finally formed first is collected. The collecting ducts you can still modify, but once it gets into the calyx, well, at the end of your duct, you can't modify it anymore. Drips into the calyx, and then you start this transitional epithelium. The minor form major, and the majors come together and form the renal pelvis, which means sink. Then it goes into the ureter, then it goes into the bladder, and then the proximal part of the urethra will also have transitional epithelium. And then it will turn into stratified squamous epithelium. So all the way from where the urine is first dripped into the collecting system down to where the urine is first exiting your collecting system, you have this urinary epithelium. Other thing that is also distinctive, it's not unique to this epithelium, but it's unique for epithelia, is that you get binucleated cells. So they think that probably this is because the smaller cells fuse to form these big dome-shaped cells. So when you section just right, you see that there's two nuclei. When you only see one, you just didn't cut through the cell probably in the right place to see that it has two nuclei. So dome-shaped and binucleated, probably you look for the dome shape first, it's going to be the most obvious, and then you can see binucleated and go, aha, confirm this is transitional epithelium. So now back to columnar. Here we've got columnar with something on top. If you're looking at a light micrograph and you can't tell how long is this, is that really, really long? Is that short? Well, one thing that will clue you in right away, whether it's a microvillus or a cilium, is you look for presence of a basal body. You can't see individual basal bodies, but if they're there at the base here, it's going to be very dark staining. It's going to pop out at you that there's something at the base of these projections, and that's your basal body. Here you don't see a big dark stripe there. So this is microvillus. Higher magnification, you'd be able to see it a little bit more clearly. Columnar in shape, but you look here, there's a nucleus there, and then there's nucleus up above. This one's a little bit below that one. It's got a nucleus down here. So you got nuclei at different layers. To be absolutely correct in naming what's on your right, you would have to say it's pseudostratified. But if you look at the illustration, the nuclei are all at the same level. So illustration, you would say it's simple columnar with microvilli. So you'd always want to be as, in lab, you get credit for being very complete. So if it says be specific and complete, give as much information as possible. So this would be columnar epithelium with microvilli. Where do you find columnar epithelium with microvilli? Stomach. And you find it in the intestine, find it in the gallbladder, collecting ducts of the kidney. Not really tall microvilli like that. When it's tall and it's really compacted, very close together, it tells you there's a lot of absorption and secretion going on. So collecting ducts, not that much, they can do it, but they're modifying. They're not really changing what's in the lumen too much. So primarily there, when you have that high density, you're probably in the intestine. What do you have protection because it's a tall cell, but you also lose a lot of these cells just because the fluid or the food is going by, the waste products are going by, and you lose them. So it's only a simple layer, so you're going to have to replace those cells. So they do provide protection, but since it's a simple layer, it's got to fill in from the side. Can do a lot of absorption and secretion because that apical plasma membrane is folded. That's what microvilli are there for, is to fold that apical plasma membrane and increase the surface area for absorption and secretion. So it says here it's 3 to 4 times taller than it is wide. That's a definition of columnar. Now you have, more obviously, you got some cells that are right at the base of the lamina. You got other nuclei that are above that, other nuclei that are even above that. But as the illustration shows, all of these cells have contact with the basal lamina. So although the nuclei are at different levels, they are not a stratified epithelium. They are pseudo-stratified. Pseudo-stratified, most places, it's going to have cilia on the surface. Here you look and you see this dark staining line at the base of these projections. That's the basal body. That's what gives it away that this is cilia and not microvilli. So you come back here and you don't have that dark line. Cilia tend to be a little bit further apart when you stain them and slap them on a slide, but they can be pretty close together in actuality. Here they're a little longer, so they're kind of feathered out at the tip. They're pretty close together at the base, and then they have this really dark basal body that is all this tubulin in the centriole-like structure. They are there for protection. They can provide protection because anything that gets on their surface will be sticking to mucus that then they can push away and actually clear out of the system. So they don't necessarily do absorption and secretion themselves, but they're a component of their epithelia is a cell that has microvilli on it that will do absorption and secretion. So cilia themselves, they're designed for movement, and they're not designed to bring things across the plasma membrane. That's not their function. But the overall pseudostratified ciliated columnar epithelium will have next to it a goblet cell. So respiratory and GI tract, they have a goblet cell, which is a unicellular mucus-secreting gland. So when you see pseudostratified ciliated columnar with goblet cell, that means you're probably in the respiratory tract or parts of the GI tract. So nasal cavity, the trachea, bronchi, all the way down to portions of the terminal bronchi. They're getting really small, so there's not too much space for all these tall cells with cilia on them. You're going to transition to other protective mechanisms and get deep into the lungs. In the male reproductive tract, you have pseudostratified epithelium, but you have stereocilia there. They are microvilli that are going to absorb the secretions that are coming out of the testes and help move the sperm out due to that absorption. So it's a cilia, but it's a stereocilia, so you've got to remember that's really a non-motile cilium, which is really a very long microvillus. Stratified columnar, more than one layer. Top layer does not touch the basal lamina, so truly stratified. And top layer is columnar, it's taller than it is wide. So this will provide more protection. Where do you find it? You're going to find it in parts of your digestive and respiratory tract that are pharynx is shared by digestive and respiratory tract. Epiglottis is covering your respiratory tract when you swallow, and then it will pop open when you're breathing. You're going to have respiratory epithelium on the side that goes down and is in conjunction with your larynx. So that will be pseudostratified ciliated columnar with goblet cells. But on the other side, you're going to have a protective epithelium that will prevent the food that's passing by from damaging that surface. Anus, mammary gland, salivary gland, urethra. They'll have components of their tract that will be this columnar, stratified columnar. Mucus, so for your own edification, so you sound really smart. Got 2 minutes and then we have to take a break. This is so cool, it beeps at me, but you can't hear it, but actually it's buzzing at me. M-U-C-O-U-S. This is the type of tissue that secretes M-U-C-U-S. So a mucous tissue secretes mucus. And it's not a typo, it's actually, this is the secretion. And this is the type of tissue that's doing the secretion. So I had some students ask me that, and like, yeah, it's a legitimate question. Is it a typo or is this something else that we're talking about? Okay, so mucous is the type of, in this case, a secretory tissue. We have a mucous membrane that will secrete mucus that will then cover the surface. So there'll be mucus, M-U-C-U-S, on the surface here that comes from these cells and also the cells down below. Usually M-U-C-O-U-S is a columnar cell. I mentioned the goblet cell. It's a unicellular gland, and it secretes mucus, M-U-C-U-S, as well. And then you'll have down here in your salivary glands, you'll have cells that secrete M-U-C-U-S, and these are M-U-C-O-U-S cells. So mucous cells that secrete mucins. So mucus cells, and part of the secretions from those cells will be mucus. The stickiness in your mouth is from these mucus cells. And then there are other cells that will stain differently. We'll see them after the break. And then you will see that they are the watery. They secrete the watery saliva. Okay, so take a break. We'll come back to this slide.